Hello everyone, and welcome to today's live broadcast, Capturing Cocaine Memories in the Perineuronal Net. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. LabRoots is the leading scientific social networking site, and we are extremely proud to bring you this interactive web seminar. For more information, visit us at labroots.com. Here's how this presentation will work today. We want to hear from you, so questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located on the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce our wonderful speaker for today, Dr. Barbara Sorg. Professor Sorg received her PhD in biochemistry from the University of Maryland. She is currently a professor in the Department of Integrative Physiology and Neuroscience at Washington State University. She is the former director of the Alcohol and Drug Abuse Research Program and currently co-director of the Translation Addiction Research Program. The main projects in Dr. Sorg's laboratory focuses on how to prevent relapse in cocaine in rats. She uses condition place preference and drug self-administration procedures to diminish cocaine-associated memories that are thought to cause relapse behavior. The majority of her studies examined the role of the structure called perineuronal nets that contribute to brain plasticity. Other studies focus on using these same models of drug addiction to determine how perineuronal nets are influenced by day-night cycle and by sleep. It's now time to present our speaker, Dr. Barbara Sorg.
Thank you, Susie, and thank you for the opportunity to share my work that we've been doing over the last few years on the role of perineuronal nets in capturing cocaine memories. Nets are shown here in green. These are these beautiful little structures that wrap around just certain neurons within the brain and the spinal cord. Today, what I want to talk with you about are three main topics, and that is the perineuronal nets, or PNNs as I'll call them, what perineuronal nets are, and why we think they are present in the brain. And then go on and talk about the research we've been doing and look at the contribution of PNNs to cocaine-associated memory. And then how PNNs themselves change after exposure to cocaine. So Camilla Golgi first described the intracellular reticulum, which we're now all very familiar with, called the Golgi apparatus, but he equally emphasized this peripheral reticulum, which we now call perineuronal nets. And he described this as a kind of corset of neurokeratin which impeded the spread of current from cell to cell. If you look at this depiction of Golgi's perineuronal nets, you can see, especially in the bottom section here, this kind of lattice-like structure with holes in it. And a more modern picture is shown here. This green part here is the perineuronal net. And that is wrapping around both the soma of the neuron and then these proximal dendrites shown here. If you look up the close-up in the lower left, you see that where the holes are located is where the synaptic boutons come in and synapse onto the underlying neuron. Now, this is a closer version of what we see in a perineuronal net. And from much work that's been done to date, we know that there are four critical components, four different types of molecules that are very important. On the left-hand side here, you can see the list of those molecules. And then on the right side, I can show you what those four molecules are. First is hyaluronan, and that is this long backbone you see in purple, a backbone of hyaluronan, just these sugar groups. And those are extruded by hyaluronic acid synthase, shown by the little square that's hyaluronic acid synthase enzyme embedded within the membrane of the neurons that extrude hyaluronan. Second is the lecticans, and these are also referred to as chondroitin sulfate proteoglycans. There's a core protein shown in green here, the core protein, and then the glycosylated, heavily glycosylated, and those glycosyl groups have sulfate groups on them. So this is chondroitin sulfate proteoglycans. The third group of proteins that's important are called link proteins. Those are shown in pink here, these pink dots, and those link the lecticans to the hyaluronic backbone. And then fourth and finally is tenascin R. Tenascin R are other proteins that bind to the opposite end of the lecticans and together form this whole lattice-like structure that wraps around these neurons. The PNNs first appeared during development, and what I've shown you here is a rat prefrontal cortex on the left and a human prefrontal cortex on the right. And just before I forget to mention, we often label these nets with, shown in green here for the rat prefrontal cortex, with Wisteria floribunda agglutinin, which is a plant lectin, WFA, that binds to these nets. And what you can see is at postnatal day 21 in the rat is there really no nets formed yet. And so it's not until late adolescence, probably 40 to at least 50 days in the prefrontal cortex, that these nets are fully formed. So on the right-hand side is the adult nets, and you can see these are really well-formed nets, very punctate kind of staining and intense staining around both the cell body and the proximal dendrites. In the human prefrontal cortex, we see the same picture. Around 20 years of late adolescence is when those nets are fully formed. Their formation is really interesting in that it marks the end of the critical period of brain development. For example, in the visual cortex, the ocular dominance, these are very important and mark that period of when ocular dominance plasticity closes. That is that window of opportunity when there's still plasticity. And so that co-terminates with the closure of ocular dominance plasticity. During development, the formation of nets is dependent on neural activity in the underlying neurons, and that's been shown with activity dependent on potassium, calcium, and the glutamate receptors, NMDA, and also calcium permeable AMPA receptors. So those all promote the formation of these perineuronal nets during development. The function of perineuronal nets is manyfold. One, they stabilize synaptic contacts. So if you look at A on the right-hand side, they form a physical barrier to new synaptic inputs coming in. So the net is shown around a neuron, and the new synaptic inputs have a more difficult time forming new synapses. And interestingly, shown on B, is these nets also capture inhibitory molecules shown in these little X's that are placed on the net. An example of this that's been studied probably the most to date is semaphorin 3A, a chemorepulsant agent that keeps new synapse formation or new incoming synapses. And then another function is shown in C here, is they've been demonstrated to restrict lateral mobility of receptors, in particular AMPA glutamate receptors. So the receptors cannot show lateral mobility, and this may decrease some excitatory activity in the underlying neurons. And third, they protect from oxidative stress. I'll come back to this topic a little bit later, but they do protect the underlying neurons from that oxidative burden or oxidative stress. And then they are also believed to regulate the ionic microenvironment. And the way they do this, if you look up in the right-hand corner where the nets are, there are a lot of negative charges that are associated with those sulfate groups on the chondroitin sulfate proteoglycans that I mentioned that make up the net. And so these negative groups are thought to serve as buffering capacity for positively charged molecules like calcium, perhaps potassium, and also glutamate. So what's exciting about perineuronal nets? Well, one of the things that's been found through the years is that if you remove these nets, you can reinstate brain plasticity, and more importantly, you can reinstate juvenile-like states of brain plasticity. So this has been shown in the visual system. I already have mentioned this with the ocular dominance plasticity. If those nets are removed in adulthood, you can reinstate that window, that critical period. They're also important in recovery from spinal cord injury. Removal of nets can promote the recovery from spinal cord injury. PNNs are reduced in individuals with schizophrenia and in animal models of schizophrenia. And this may be related to the fact that perineuronal nets largely wrap around parvalbumin fast spiking neurons that control output in the prefrontal cortex in particular. And so these may be important for the loss of cognitive ability in individuals with schizophrenia. And then finally, they've been shown in different studies to be important for learning and memory. And I'd like to point out that not only could they prevent certain types of promote certain types of learning, such as extinction learning, they may also prevent certain types of learning induced by other stimuli. And that's where our studies came into the picture with cocaine, because cocaine is a strong stimulus that induces its own learning and memory. So we asked the question, what about drug memories? And in particular, memories that we believe drive relapse behavior. So what is the contribution of perineuronal nets to cocaine-associated memories? So we have to think about what these drug memories are. So first, we think about accumulating these drug memories, or memory for how the drug would make one feel. And that would be including the euphoric effects shown here on the top, and then also on this bottom. This is supposed to depict the withdrawal effects, the negative effects, and maybe anhedonic effects of drugs. And so these internal cues that are induced by drugs, first when they come on, and then when the drugs come off and the withdrawal effects ensue. And so these are partly creating these memories. And then there's a memory for the drug environment. So this would be the environment where one might take drugs or would get their drugs. And there are memories for these drug cues. So the very discrete cues that become associated with taking these drugs. So I've kind of written this as just a composite drug memory in the center here. And all of these, we believe, contribute in a large part to the motivation to continue to use the drugs and to relapse in taking these drugs. So our focus then is on how to diminish those cocaine-associated memories. To think about how to diminish memories, we have to take a look at how memories are consolidated and reconsolidated. And in other words, how they're acquired when they're initially consolidated somewhere in the brain, likely many locations in the brain, and then also how these memories are maintained by the process of reconsolidation. So to look at these processes, or just take an example of a drug experience that is consolidated or stabilized somewhere in the brain or in multiple parts of the brain, and I just call this a stored drug memory. Now we know that stored drug memories aren't forever. They can be retrieved and they can be altered in some way. So when these memories are retrieved or recalled, or as I'll later say, reactivated, every time these memories are retrieved, they are susceptible to strengthening or perhaps weakening or updating. And this is because these memories for a certain period of time, probably several minutes to hours, become unstable and therefore labile. So they could either be strengthened, they could be weakened, or they could be updated. And that's by the process of reconsolidation, where that memory could be restabilized back into the stored memory state. This reconsolidation process requires molecular events, and in fact, probably several molecular events. Many of these have been explored in the last 15 years or so. And the fact that there are several molecular events means that you could potentially block some of these events and reduce reconsolidation or block reconsolidation of memory, and therefore reduce that stored drug memory in some fashion. And we don't know if blocking reconsolidation reduces the stored memory or impairs the ability to retrieve that memory. And that has not been determined yet. Regardless, the idea is that we could take a stored drug memory and potentially block it in some way or diminish it in some way so that we could decrease relapse. And that would be our ultimate goal. So we can think of this as, this is one example I like to give of updating memory. So we're always updating our memories and in some way either enhancing or diminishing or updating and changing them out in some way. So if you think of when you were small, you think of the word president, and the president conjured up this image, probably of George Washington, and later on maybe talked about Abe Lincoln. And through the years, you've updated this memory to think about what president means to you. And so depending on your age, and this is probably showing my age here because I think of this president first in my memories, and through the years, this has been updated for better or for worse through the years. And here's where we are now, of course. So coming back to the circuitry involved in drug reward and thinking about where these perineuronal nets may be important, I've been focusing my work in the last several years on the medial prefrontal cortex here. So this is a rat brain, a sagittal section of a rat brain, showing you lots of connections that are important in this circuitry that's important for rewards. So my focus is on the medial prefrontal cortex, and so we'll dig in a little bit deeper here, is showing now a coronal slice of the medial prefrontal cortex in a rat in this area here that we're interested in targeting for our studies. The reason for this is that there are pyramidal neurons in the prelimbic region of the prefrontal cortex that have important outputs to the nucleus accumbens for relapse behavior. So we think of relapse behavior in individuals, in drug addicts. We measure this in animals, but we call it reinstatement. So I will be talking much of this, the rest of this is talking about reinstatement rather than relapse. So we know these outputs are very important from some previous work that's been done, showing over here on the right side. But one of the things we're very interested in is what's controlling those pyramidal neurons. Those are very important, again, for relapse, but there's an upstream control of those neurons, and those are by these GABAergic interneurons located in the prefrontal cortex. Those contain parvalbumin. They're fast-spiking neurons, and they can powerfully inhibit the output of pyramidal neurons. Now, the reason that we're very interested in these nets, in these neurons in particular, is that 75 to 80% of these GABAergic parvalbumin neurons have perineuronal nets surrounding them. And so we're interested what the role of these nets is in this reinstatement behavior. And so what I'm showing you here in the lower left is the nets, these perineuronal nets labeled in green. And then in the middle panel is parvalbumin in red, and then the overlay. So you can see that there are several parvalbumin neurons that are surrounded by nets. So we're looking at what is the contribution by perineuronal nets in the prefrontal cortex to the cocaine-associated memory. For that, we used a couple of different animal models of addiction. One of these is called the conditioned place preference model. And most of you should be familiar with conditioned place preference. If you like to drink coffee in your workplace, you may go to the place where they make coffee or where you make coffee every morning. You've been conditioned to prefer that place because it contains something you like. And so what we do in the rat model is we take a rat and we put that in a 3-chambered box, a white side, a gray side, and a black side, typically. We'll put the rat in the box and ask it where it would like to spend its time. So the animal is kind of getting an initial preference, and it'll wander around the box. It's deciding where it likes to be. So a couple of days later, we take that same animal and we give it an injection of cocaine, try to recreate the environment of cocaine, and then we confine it by closing the door and letting the animal experience this cocaine feeling, the internal sensations of cocaine, for about 20 or 30 minutes. The next day, we take the animal and we put it on the opposite side and give it a boring saline injection and confine it to the other side. So this has no psychostimulant effects the way cocaine does. And so we train the animals day after day, alternating between the cocaine side and the saline side. And then on the final test day, we take the animal, put it in the center, and ask where it would like to spend its time. And most animals inevitably will go spend their time on the cocaine-paired side. So what we're looking at is how much time they spend here initially and how much time they spend here after they've been trained. So we come back to the question, are perineuronal nets surrounded cells activated by a cocaine-associated memory? So that was our first question. And this was done by Megan Slaker, who is now a postdoc at the Medical College of Wisconsin. And Megan was very into her PhD project, so much so that at one of the Halloween parties, she showed up dressed up as a perineuronal net herself. So here's what we did. This is what Megan did, was she trained the animals, as I just mentioned, cocaine on one side, saline on the other side. And then after a period of time, what she did after they were trained was she confined the animal to either the cocaine paired side or the saline paired side. And so this would reactivate the memory on the cocaine side. And that was the idea. And then a few hours later, she measured what happened in the perineuronal nets, and she looked at the intensity of these nets, and she also then looked at the number of neurons that are surrounded by nets expressing CFOS, which is a marker for activation of neurons. And this was all in the prelimbic cortex. What she found was she measured the CFOS cells that were surrounded by perineuronal nets, and you can see in the white bar, these animals were confined to the saline side, but the blue bar shows the animals confined to the cocaine side, and that you see a greater activation of CFOS neurons in the prefrontal cortex that are surrounded by perineuronal nets. So it looks like a cocaine memory does activate these neurons that are surrounded by nets. So next, she asked the question, could she disrupt consolidation of the drug memory, and the cocaine memory in particular? Do we need perineuronal nets there? And secondly, do we need perineuronal nets for the reconsolidation process? So what she did was she used the most common way to destroy nets, and that is to give this enzyme chondroitinase ABC, or CHABC. And what I'm showing you here on the lower right is that these nets can be disrupted with chondroitinase ABC. This is an enzyme, and this happens within probably a couple of hours. We usually test animals days later because this lasts for a fairly long time, and I'll get back to that issue about how long this lasts. So she disrupted these perineuronal nets. This is before the enzyme on the left side here. We use our Pac-Man to depict enzymes, the CHABC enzyme, and then afterwards, just 3 days later, this is what it looks like. So you might see an occasional net. I don't know if you could pick one out there in the right-hand side. You see very lightly stained nets or none at all 3 days later. We've also, I'm not showing you the data, but CHABC does not cause underlying cells to die. This has been shown by us as well as other labs. So what she did was she said, well, let's give this vehicle, just vehicle or CHABC to the animals in the prelimbic prefrontal cortex, and then train them, as I mentioned before, and then test them. And so what I will show you is the results of that test day. On the left side is shown the behavior, and what you're looking at on this y-axis is the time spent in the cocaine-paired side. So if you look at the initial preference day, the white bars show you that in vehicle-treated animals, they show more time they spend on the cocaine-paired side after training. But you see a reduction of this, the blue bars, after CHABC, showing that perineuronal net destruction does not allow the animals to acquire this memory as well. On the right-hand side is shown the double-labeled cells, the CFOS neurons surrounded by perineuronal nets that's labeled by WFA. So these double-labeled cells are also reduced. So it mirrors what we see with behavior. And then also, Megan also did some studies to show that a nearby brain region, if you look at the upper left panel here, she went just ventral to the prelimbic cortex down into the infralimbic cortex to see if a nearby site would affect the memory the same way, and she found it did not. So you can see in the bar graph here that the CHABC treated animals do not have their memory affected, so they test just fine. Then she wanted to test for memory reconsolidation. So I wanted to show you how we test for memory reconsolidation. And that is to go ahead and train the animals for this condition-placed preference, or CPP. And then after a period of extinction, and what this is we basically place the rat in the box day after day until they no longer show a preference for the cocaine-paired side. So we're kind of overriding this memory and extinguishing that response. And then we give the vehicle or the CHABC into the prefrontal cortex. And now we have to reactivate the memory, just like I showed you earlier. We have to reactivate that memory to make it become labile for a period of time. The way that she did this was to give a cocaine injection, and essentially it's a reminder of what the cocaine felt like. And the animal will spend more time on that white side of the box again. And then at a later date, and this was several days later in this case, she reinstates the memory. And the way we do that is, again, to give a cocaine injection. It's another reminder. So our question here is, do the rats maintain that cocaine memory once it's been reactivated in the absence of NETs? And so on the top is showing you pretty much the same thing I just described. If you look from left to right, there's the CPP training that she did, the testing and the extinction that I mentioned, then the injection of the ABC into the prefrontal cortex. And then she either reactivated the memory, as I said before with cocaine, or she had another group that received no reactivation as a control group. And then after a period of time, she did some more extinction or just let them sit in the home cage and tested on the last day, the day shown here, the reinstatement day, whether the memory was maintained. So first I'll show you the results of animals that had their memories reactivated. And that's shown here on the left side is the behavior. We see very much the same thing as we saw before for consolidation, and that's that CHABC treated animals, that is animals with no NETs, show a diminished behavioral response compared with their controls. So it looks like they're not reconsolidating the memory or maintaining their memory as well as controls. We see that mirrored in the CPOS activation in the prefrontal cortex. So these are CPOS neurons surrounded by perineuronal nets. And there's a decrease in the activation of those. Of course, we have to know that the treatment with CHABC by itself does not somehow just impair memory, whether it's reactivated or not. So we have a control group of no reactivation. So that is, these animals received vehicle or CHABC in the home cage, and that's it. And then they were tested later for reinstatement. And that group is shown here. So when we don't reactivate the memory, we see no changes in the responses for behavior. So they condition place preference just fine. And we see no differences in the CPOS activation. So it appears then that giving or removing NETs, even after training, can prevent the maintenance of those memories. And one thing that we wanted to check was, or that I would like to show you too, and we checked, was how well were these NETs reformed 2 weeks later, because this is the period of time between when the CHABC was given and when the animals were reinstated. And that's shown here. So on the left side is shown vehicle-treated rats, both with a lower magnification and a higher magnification, shown on the left side. And then I showed you before what happens after CHABC. This was 3 days later. You can see very few, maybe one net, very lightly stained. And then you can see about 2 weeks later, these nets are starting to reform. But you can see that the nets, if you look at the higher magnification on the right-hand side, those nets have not been fully reformed, and they're very thin and not very intense, and really generally not wrapped around the dendrites. And then this is a quantification of that. So 9 days later and about 2 weeks later, we still see that the intensity of the nets has not been restored to normal. And we believe that the intensity of the net matters, and that will, I'll be alluding to that. We're talking about that in a few minutes. So then secondly, we did a few studies. We're just beginning some of these studies using the gold standard for addiction models, and that is a self-administration model where the animal learns to press a lever to receive cocaine in our case. So this animal has a tether. You can see this tether on the animal. This is an IV line that's hooked up so that when the animal presses a lever, it gets a small amount of cocaine. The other thing is when the animal presses the lever, and it's hard to see in this picture, but it's just below his nose. He looks like he's pressing it with his nose on his chin, is that there's the cocaine lever is shown here. And then above that lever, every time the animal gets cocaine, it also gets a cue light above the cocaine lever. And the animals they learn to pair this cue light with the cocaine, or the feeling of cocaine that they get with the cue light, so that at a later point in time, once they've learned to press that lever for cocaine, they will press the lever just to receive the light. So it would look something like this. This is just sort of an average self-administration protocol. And on the left side is shown, on the y-axis is shown the responses. These are what we call active lever, the lever that gives cocaine. It's usually another lever in the box that gives nothing at all, called the inactive lever, shown at the bottom in green here. So the animal will fairly quickly, depending on the dose of cocaine, will fairly quickly learn to press the lever for cocaine and maintain a certain level. And then you can take the cocaine away from the animal. And it's usually quite a surprise the first day. On the first day of extinction, the animal will press the lever more in a desperate attempt to try to get some more cocaine. But they will also fairly rapidly extinguish this response, and they learn not to work for nothing. So they press the lever and get nothing during extinction. And then what you can do is to give that cue light again, just a single, what we call a priming cue light. It just primes the animal to start pressing the lever again. The animal is not receiving cocaine, but it will continue to press the lever for a period of time just for the cue light. And this is the reinstatement of that behavior that was extinguished. This is our equivalent of relapse in animal models. So this is what we saw when we asked the question, do we need perineuronal nets for the acquisition or this initial consolidation of cocaine memory when they're learning to press a lever to get cocaine? So on the y-axis again is showing these active lever presses, and you can see in the blue, the blue line is showing what happens in vehicle rats. They acquire this habit, or they acquire the lever pressing behavior. It takes about 10 days to show kind of a maintenance level or a steady level of self-administration. And you can see that in our animals that have no perineuronal nets, this is a slower response and it never quite catches up to the vehicle controls. So again, we don't completely blunt, but we do seem to blunt that acquisition or consolidation of that cocaine-associated memory by removing nets in the prefrontal cortex. And then we also we've just begun looking at reconsolidation using this self-administration model. And for this, it's a little bit, takes a little explanation, but if you look at the left-hand side of this timeline at the top, the self-administration training is said here in FR1, and that means that every time the animal pushes the lever for cocaine, it gets a single injection of cocaine, a fixed ratio 1. So push once, get a cocaine injection. And we do this for 10 days, and after that period, then we inject CHABC or vehicle in the prefrontal cortex to remove nets. Wait a few days, and then here, what we were doing on where it says reactivate and BR5 in red, what we were attempting to do was to update the memory. In this case, the animals have pushed that lever hundreds of times under that so-called FR1 schedule. They're used to getting a cocaine injection every time they push the lever. In this case, we essentially trick the rats and say, well, you need to get out of that sort of habit responding, pushing that lever and expecting a cocaine injection. And instead, we're going to fool you a little bit, and we're going to make you press maybe once, maybe twice, maybe 9 times, an average of 5 times on a randomized schedule. So basically, the animal has to learn a kind of a new protocol. And so it doesn't quite know what to expect. And here we think we're creating an error signal. This is some work that was done a few years back by Jonathan Lee, showing that to update a memory and therefore make it labile again, we may need to kind of trick the animals and make them update their memory. So this is our attempt to update the memory, is to give the animal a 30-minute session to reactivate the memory, but again, under this variable ratio 5 schedule. And then the next day, we give them a very short extinction session, and then we give them that cue priming, where we give them one light stimulus, and then we let them go to town. They can press the lever as much as they want during these last sessions. What we found, I'm just showing you first what we found for the last 3 days of training. No one has been treated yet with the CHABC, so we expect animals to be the same. This is just showing the last 3 days of training, days 8, 9, and 10. Animals are pressing the lever for cocaine. Then we reactivate the memory, and you can see that they show the same lever pressing behavior during reactivation. Whether they have nets or not, they're still pressing the same. Then we give that extinction session, and we also saw no differences here. But when we give them the cue light again, the vehicle animals show a nice increase, as expected, to press just for the cue light, because that has become associated with the feeling of cocaine. However, in animals that have their nets removed, we see no increase in lever pressing behavior. We suspect this is probably a disruption of the reconsolidation, but we are really at the beginning of these studies, so we would like to continue to pursue this. So in summary then of these data, we believe that for the consolidation process of this cocaine-associated memory, we need intact perineuronal nets, and we also need them for the reconsolidation process to occur. So that's what I've shown here. One of the issues, though, is that we've got, we give this enzyme, this kind of artificial situation where we give an enzyme to the animals to destroy their nets. So we wanted to kind of flip the question on completely around and ask the question, well, what happens in intact rats? What is the impact of cocaine exposure itself on perineuronal nets? And in particular, we were interested in the intensity of these nets because we believe that the intensity of the nets matters. So how do these perineuronal nets change after cocaine exposures? There have been some studies, most of these have been done just in the last couple of years, ongoing studies, and while we're continuing ours as well, and they found that PNN intensity is altered after cocaine, after ethanol, and after nicotine. And I won't go through the details of this, but I put the papers up here for your interest so you can take a look at these. But the bottom line is these nets have been shown to be much more dynamic than originally thought, and they appear to be, the changes appear to be dependent on the drug itself, on the class of drug, on the amount of drug, on the withdrawal time, withdrawal times from the drugs, and also the brain area that's been examined. And here you see the cerebellum, the insular cortex, orbital frontal cortex, and VTA have been examined. We were particularly interested in what happens in the prelimbic part of the prefrontal cortex early after cocaine exposure. And so we also looked at this parvalbumin expression because, as I said, most parvalbumin cells in the medial prefrontal cortex are surrounded by nets. And parvalbumin expression may be very important for the firing of these very fast spiking neurons. So we did the simplest experiment possible first by just giving either a saline or a cocaine injection, and then waiting 2 hours or 24 hours later and looking at what the intensity of the nets were and the intensity of parvalbumin neurons underlying those nets. And then secondly, and that picture is shown here, the red staining for parvalbumin, and the green again, the perineuronal nets surrounding those cells. And secondly, we looked at 5 days of exposure. This is not a lot of exposure, but there have been a number of studies that look at just IP cocaine injections. And so we thought, well, perhaps we could compare some of our studies with previous work that's been done. So we gave 5 cocaine or saline injections and looked 2 or 24 hours later for perineuronal net staining and parvalbumin staining. And in general, what we found, if you look at the top, is that 1 day of cocaine decreased the intensity of parvalbumin and perineuronal nets, whereas 5 days of cocaine produced just the opposite response. It increased the intensity. So looking at the graph, if we look on the far left, this in the green, we see that compared with controls, there's a decrease of, it's not a huge decrease, but it's a significant decrease in the intensity of the nets 2 hours after a single cocaine injection. And this is returned to normal 24 hours later. If you look in the second graph here, after 5 days of cocaine, 2 hours later, we don't see a decrease anymore. And in fact, we see an increase in the intensity of those nets, perhaps the thickness of those nets. And that returns 24 hours later. For parvalbumin, we see largely the same picture. Everything's just perhaps a little bit delayed, but it pretty much mirrors what we see with these perineuronal nets. So we see some pretty dynamic changes that happen within 2 hours, but as you can see, they're in opposite directions depending on whether it's just a single cocaine injection or 5 days. Interestingly, we and others have found that the intensity of the perineuronal nets shown on the y-axis is correlated fairly tightly with the parvalbumin intensity. And as mentioned, this is found by others and by some of our studies. And we don't know what this means, but it could mean that these molecules, or at least some molecules in the net, along with parvalbumin, are co-regulated in some way. And this has not yet been determined. So what we find in summary of these studies is that in naive animals, we see a certain thickness or certain intensity of the net. In animals that receive 1 day of cocaine, we see a decrease, and so I've shown this as a decrease in the net here. And we believe that this decrease in intensity could allow for the formation of new synapse formation. As I mentioned early on, those holes in the net appear to, or the structure of the net appears to prevent new synapse formation. So if you make that net thinner, you perhaps allow new synaptic inputs to come in, and therefore potentially consolidation of new memories, in this case, cocaine memory. And then after 5 days of cocaine, we see the opposite response. We see what we think is a thicker net, or at least a more intense net. And in this case, this may not allow for new synapse formation, and in some way allow for the consolidation of strong memories. And that is perhaps we have kind of locked in, if you will, these cocaine-associated memories. One of the things that we're interested in is looking at this new synapse formation in particular. We have some early studies that just show synapsin 1 labeling, so just to look at general synaptic inputs. We don't know if these are true synapses, but synapse 1 labeling increases in animals after CHABC treatment, and this was about 2 weeks after CHABC treatment. We're also very interested in pursuing what kinds of synapses these are, if they're glutamatergic, shown on the left here with this labeling with vGlut1, or GABAergic labeling. And these studies have been done by Dr. Lynn Churchill, also at Washington State University, and ongoing collaborative work with Dr. Sue Eicher at Oregon Health and Sciences University. So then we just have started to look at the potential mechanism for changes in intensity. And for this, we've looked at oxidative stress as a mechanism. And the reason that we did this was that there's some evidence that perineuronal nets protect their underlying cells from oxidative stress. And secondly, that cocaine itself induces oxidative stress through its generation of reactive oxygen species. So we came back to the simple question and asked, what does 1 day of cocaine exposure do to the nets and oxidative stress marker? So 1 day of cocaine, we know, will increase dopamine. Dopamine, we do know, can increase reactive oxygen species and increase oxidative stress. So what we did was to give an antioxidant, N-acetylcysteine, or NAC, and we, from many, many studies, know that this increases glutathione and will decrease oxidative stress. So we gave this NAC or vehicle a couple of hours before they received a single saline or cocaine injection. And then again, we waited this 2-hour window and looked, in this case, at staining for perineuronal nets, for parvalbumin, and then also 8-oxo-DG, which is an oxidative stress marker. And so what we shown on the left panel are the photomicrographs of that labeling. And we were interested in looking at 8-oxo-DG neurons that are parvalbumin neurons surrounded by nets, as well as those that were not surrounded by nets. So looking at the right-hand upper panel, what you can see is if you look at the white bars, we just normalized everything to saline vehicle, animals that receive saline and vehicle. And then you can see in the black bar that cocaine does increase the oxidative stress marker a little bit by about 20%. And this is prevented by pretreatment with the antioxidant NAC. what we see in parvalbumin-containing cells that do not contain nets is a different story. This is also an ongoing study, but so far what we see is this increase in nets, and it seems to be that we've got cumulative effects or potentially additive effects of both cocaine and NAC, shown on the far right bar. And we're not quite sure what's going on. We have some ideas, but clearly the parvalbumin cells without nets look very different from the parvalbumin cells with nets. So overall, it appears so far that these perineuronal nets protect against cocaine-induced oxidative stress. We also are just at the beginning of some electrophysiological studies, and this is done in collaboration with Dr. Travis Brown at the University of Wyoming. And what he's done is to give saline or cocaine injection, again as before, wait 2 hours, and then look at prefrontal cortex self-firing, and in particular looking at these fast-spiking interneurons that contain nets, as well as fast-spiking interneurons that do not contain nets in this deep layer of the prefrontal cortex in layers 5 and 6, where the pyramidal neurons have their outputs to the nucleus accumbens. What he's found so far is pretty clear, and that is that these fast-spiking neurons that do not contain nets show fewer spikes. So on the left-hand graph, it's showing on the y-axis the number of spikes as the current is increased. And you can see in the open symbols, saline-treated animals, there's an increase with current and a decrease in that ramping up. So basically, cocaine treatment just a single day and 2 hours later decreases the number of spikes. However, you see on the right-hand side, the same number of spikes, in other words, no differences between saline and cocaine-treated animals. So it appears that PNNs prevent the cocaine-induced decreases in the number of spikes. So again, there's a lot of work to do. We have a lot of questions ahead. But we're really interested in how these neurons are firing differently, and then again, in how these neurons might be different when we remove nets. These are all, as a reminder, these are all intact animals. So in summary then, we have the naive animals, as I showed before. We have potentially extra inputs, extrasynaptic inputs coming in after a single cocaine injection, the decrease in the nets, the increase in oxidative stress. But the consequence of this, we believe, is that we may enhance cocaine-induced plasticity because of the added inputs that are coming in that may be specific for cocaine-associated memories. And then again, after multiple exposures to cocaine, we see potentially this inability for new synapses to form. This is something that we're currently measuring. An increase in PNNs, which may be some sort of protective response from oxidative stress after multiple injections of cocaine. But the consequence of this would be that these neurons may be resistant to new plasticity. And this may be akin to what has been described many times over in the addiction literature, and that is that natural reinforcers do not seem to induce the plasticity once individuals or animals have been exposed to cocaine. And so this resistance to new plasticity may in part be occurring at the level of these GABAergic fast-spiking neurons. And finally, and I would like to thank all the individuals in my lab who have generated this work, collaborators at Washington State University, the University of Wyoming, and Oregon Health and Science University. And I would also like to thank our local alcohol and drug abuse research program, as well as the National Institute on Drug Abuse. And I would like to thank all of you who are participating in this seminar. Thank you.
Thank you, Dr. Sorg, for that fabulous presentation. We want to get right to the questions and input, so here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. We will try to get to all of you, but if not, we will be sure to follow up after the broadcast. So let's take our first question. Dr. Sorg, how long does it take PNNs to return, and do they wrap around the same neurons?
So we have some, we have some idea of how long these take, but there are many questions that remain. What we've tested so far is the perineuronal nets. We've tested out to 1 month later with the dose of the chondroitinase ABC that we deliver, which is a moderate dose relative to many of the studies that are out there. Some have shown that it could take up to 2 months to reform these nets fully. We do not see quite a full recuperation of these nets 1 month later. And we do believe that the intensity of those nets matters because there have been, for example, knockout mice that have one of the critical proteins, a link protein that I mentioned earlier. When they're knocked out, those nets are still there, but they're very thin and not very well formed. And in those animals, when those have been knocked out in the visual cortex, we see, or I should say James Fawcett's lab, they've observed that those animals are still maintain that plasticity of as juvenile-like states of plasticity. So we believe that those fully formed nets are really critical for the sort of closure of that plasticity period. In terms of how whether or not they form around the same neurons, no one is sure yet, and we would very, very much like to answer that question. I believe they probably are formed around the same neurons because there are certain proteins that are produced within the neurons that have nets around them, and we believe that the expression of those genes is absolutely necessary for those nets to form around them.
Here's another great question. What is the translational value of removing PNNs? For example, could you use CHABC in humans?
So CHABC has not yet been used in humans, to my knowledge. There is some work also by James Fawcett looking at spinal cord injuries and the ability to use CHABC in, in a long-term, say, in adenovirus or in kind of hydrogels that would release CHABC for the long term and allow the kind of this reinstatement of plasticity, new synapse formation, and dendritic growth. But so far that has not been used in humans. One of the things that we're very interested in is targeting just the nets. I did not mention that the CHABC actually destroys for a period of time all of the extracellular matrix, and we know that extracellular matrix molecules are really critical for all kinds of plasticity, including cocaine-associated memories. So one of the things that we're interested in is just targeting one of those proteins, in particular the link protein. So we're just now getting underway with some behavioral studies to look at whether we can down-regulate or in some way knock down just that protein, just that link protein, and knock down only nets, but not destroy the surrounding extracellular matrix. So in that way, get us closer to a sort of a translational picture in where we might understand what components of the net are really critical, and then how we might better target just those nets to be able to bring that to from bench to bedside.
Thank you, Dr. Sorg. We have time for one more question. How does removing PNNs prevent memory reconsolidation?
We don't have a good answer for that yet. One of the things that's really interesting is that we can remove nets after animals are trained on cocaine, and we see that we get the full expression of memory. That is, during that reactivation session, whether it was for conditioned place preference or for self-administration, so either of our models, we found that when we reactivate the memory, the animals look normal. That is, they don't have a diminished response or diminished expression of conditioned place preference behavior or pushing the lever for cocaine. It's only later when we test. So the cocaine itself that we give during reactivation and or in combination with the memory reactivation is somehow initiating a series of responses, and this could be the new synapse formation, those responses that lead to the maintenance of memory. And so in the case when there are no nets, we cannot maintain that memory at a later time or when it's tested at a later time. So for example, a good example of this is just thinking about when we reactivate the memory. Perhaps reactivation of memory allows for new glutamatergic synapse formation onto those parvalbumin interneurons that then inhibit any subsequent behavior. So they would inhibit the output of the prefrontal cortex and therefore inhibit that reinstatement behavior. And that's what we've seen. What we need to do is go in and measure what is happening with that new synapse formation. It may happen very rapidly within a few hours. So those are the kinds of questions that we need to address.
Thank you, Dr. Sorg, for your fabulous presentation. Do you have any final comments for our audience?
Well, thank you. I just think this is a wonderful format to present and to allow a number of individuals to participate. I'm looking forward to attending the seminar myself. So thank you for the opportunity.
If we weren't able to speak directly with you today, we'll be sure to follow up with you. Thank you for taking part. We want to thank today's presenter, as well as joining us here at labroots.com, the leading scientific social networking site. For more information, don't forget to visit us at www.labroots.com. Today's webcast will be available for on-demand viewing on our website through 6 months from live date. We will let you know when it's posted, and we hope you pass this information on to your colleagues who couldn't take part in today's broadcast. See you next time. Goodbye.